How do enzymes work? An enzyme is a large globular protein that acts as a biological catalyst to speed up chemical reactions. It does this by lowering the activation energy required for a reaction to take place. They have a specific active site that binds with a specific substrate. In their inactive form, enzymes are known as zymogens and are activated by cofactors. One example of this is the conversion of trypsinogen via the enzyme enteropeptidase to trypsin. This can be found in the pancreas. Enzymes provide an alternative path for reactants to form products by lowering the activation energy of a reaction. They bypass higher energy transition states in favour of a lower energy transition state. Think of the underground tube station. Trains avoid the busy traffic above for a quicker journey. A key feature of enzymes is their ability to be reused after a reaction has taken place. Enzymes have a specific active site to which a substrate binds, forming the ES complex. When the two bind, this is reversible due to weak bonds. Products are released when the reaction has finished. However, this is irreversible. In the human body, the enzyme amylase can break down complex carbohydrates such as starch into simple sugars. As starch progresses further through the digestive system, glycosidic bonds are hydrolyzed repeatedly until glucose is formed. The lock and key theory was the first proposed idea of how enzymes work, however was too rigid. The induced fit theory, on the other hand, is more flexible. A conformational change in the shape of the active site is induced so that substrates can bind. The chemical structure of the substrate and enzyme bring about the specificity of binding and enhance the breaking and formation of bonds. Enzyme activity can be influenced by inhibitors. Competitive inhibitors compete for the same active site and block the substrate from binding. This is reversible if the concentration of substrate increases. Non-competitive inhibitors bind enzymes away from the active site and cause a conformational change. However, the reaction still takes place, just at a slower rate. Uncompetitive inhibitors bind at the same active site as the substrate but render the enzyme inactive. Several factors influence the activity of enzymes, such as substrate and enzyme concentration, pH, chemical changes, coenzymes, the phosphate group, and temperature. These factors all enhance activity until an optimum point. Enzymes are present and used in everyday life. One example is the use of yeast in fermentation. Yeast produces maltase, which breaks down maltose into glucose molecules. Enzyme deficiencies can lead to metabolic disorders. For example, lactose intolerance is due to a lack of the enzyme lactase.